Hi everyone, today we're going to do a pH lab. It's going to use the red cabbage indicator, which I'll describe in a minute. But basically this is a very simple experiment to learn about pH and to encourage scientific reasoning. So, we could just use a pH probe to measure the pH of a certain solution, but this way, although old-fashioned, really encourages thinking. So we're going to be using a colorimetric assay. So a colorimetric assay is basically an assay or a test that uses color to make a determination about something, in this case pH. Okay, so our data that we will determine today is color change of solution. So in that way it's qualitative and not quantitative. So unlike a pH meter, it's not going to give us the number of the pH. Rather, the solution will turn a certain color. So you'll have to determine what the color is, and you'll have to use a predetermined standard to compare that color change to. Okay, so in that way it's qualitative and not quantitative. So we will use this standard for comparison of the unknown. Let me just explain what I mean as far as this simple experiment goes. We're just going to collect a bunch of test tubes. I will have the red cabbage indicator ready for you, and we'll do the experiments. So you grab these test tubes. You'll take some red cabbage indicator, which turns out to be is purple in color, and you'll mix them with various unknown pH solutions, and a color will be created, and you'll compare that color to the standard. So how do you make red cabbage indicator? Well, you basically take these, the leaves of the red cabbage, which is actually purple, and you boil them down into some water. And the water that you collect is actually sensitive to pH and will change colors based on the pH of the solution. Okay, so the first thing that we do is establish a standard color scale. Now I will demo this for you in class. There are also videos online from previous years that show how to do this. So if you're missing class, you can go to that video and watch the process. But essentially what it is you take a test tube, you add buffer of a known pH. So I will create solutions of buffers. They will range from 1 to, I think I have 13. In the example shown here, I have a buffer with a pH, known pH of 1. These buffers are clear, okay, so it's the powdered buffer in distilled water, so it's a clear solution. Into that known pH buffer, I'll add some of the red cabbage indicator, 3 mils of that red cabbage indicator, and depending on the pH of that solution, in this case 1, it's going to turn a certain color. So for example, if I add red cabbage indicator to a known pH of 1 buffer and mix it, the color is going to turn a red. So from that I can determine that pHs of 1 turn red. So in previous years we've done this very thing, and you can see the range there from, in this case, 2 to 11. And down on the acidic side around 1 and 2, you are red. And as you become higher in pH, so as you drift towards neutral, you go from like a magenta to a pink to a purple to a darker purple. And then around neutral, you start to turn more into the bluish greens. So as you can see from this standard, pH of 8 is sort of like a turquoise green, 9 is a darker green, 10 is a lighter green, and what's not shown here is as you get into 12, 13, 14, it turns into a yellow. So if you watch that video, that's great. For the purposes of this experiment, we're going to use this artificial standard scale for our experiments today online. Okay, so it's sort of challenging. These the pens that I have, the color of the pens that I have here are rather limited, but for this experiment we're going with a standard color scale shown here, 1 being red, 3 being pink, 5 being purple, 8 being blue, 10 being dark green, 11 being light green, and 12 being yellow, darkish yellow. Okay. So in this case, a standard the standard is like a control in that we're using it for a comparison to the unknown. So we have known pH solutions that we'll use to determine that standard. So we'll compare the unknown to those known pHs. So we don't know what color the unknowns will change, but we have already established the color of the unknown, so we can associate certain pHs with certain colors. Okay, so the experimental test tubes will contain those unknown samples. In this case, for the following experiments, the independent variable is the type of solution. So you're testing unknown experimental solutions, and you're comparing them to the control solutions of known pH. That is the buffer solutions. Okay, so in this case, the dependent variable for this series of experiments, so in other words, what depends on the type of solution that you give? Well, it's the color change. Remember, this is a colorimetric test, so we're testing to see how our experimental unknown solutions change in color. So the color tells us something about this particular mystery unknown solution. So here are a couple of examples that I'll ask you to think about as we go through. I'll describe them here. You can pause the video at each of these questions and give your answer on the document that I've included with you. So in experiment 1, we're testing ammonia. So we have a solution of ammonia. We add our indicator, and the result here is a light green. If you compare it to the standard scale, our control, you could tell me something about the pH of ammonia. Experiment 2, baking soda. So you add baking soda to a test tube, then you add the indicator, the color turns into blue. So if you compare that to your color scale, what is the pH of baking soda? Experiment 3 is Coca-Cola, which is brown. So you mix it up, you get sort of this suspension of a pinkish red. So it's like a magenta. So you compare that to your results, and this is the first sort of funky result that we get. What do you make of this? Your result is sort of a magenta color, but it's also kind of brown. So what is the potential problem associated with testing Coke? And I guess I can make that extension to testing anything in a colorimetric assay that is already colored. Experiment 4, you test 7-Up. 7-Up is clear. You add your indicator, and the result is a pinkish purple. So where does pinkish purple fall on your color scale? Which brings us to a question. When you have a set scale, and in this case you have a 1, a 3, a 5, an 8, a 10, 11, and a 12, those are certain defined numbers. We don't have every pH tested here. So what do you do when you get an in-between color, as we did with 7-Up just now? Experiment 5 is vinegar. You perform your experiment, and that's the color you get. Here is your color scale for comparison. Milk of magnesia we talked about when watching the video on the pH scale. Milk of magnesia is often taken to combat heartburn. So you do the experiment, milk of magnesia comes out to be that color. Where does it fall on your scale? Experiment 7 is bleach. You perform the experiment and you get a very bright yellow. Where does very bright yellow fall on your scale? You also want to test hair straightener. You do the experiment and you get a kind of darker yellow. So where does hair straightener fall regarding pH on your colorimetric comparison scale. Last, I'd like you to consider the following, and all this will be in the Google Doc that I share with you, but hair straighteners are an interesting product. So the thing that the hair straightener does is in the title, it straightens your hair. So do some quick research. How does hair straightener straighten hair? And what I'd like for you as a last answer here, provide a biochemical description of how these products work given the pH of hair straightener. Okay? Thank you.